Hello, everyone. Welcome to this lecture for the first chapter in the Campbell biology textbook for general biology. Chapter 1 covers evolution, the themes of biology and scientific inquiry. As per usual, just an introduction into what biology generally is. Now, in terms of what we're going to be looking into, it'll be just primarily how do different organisms change with their environment to become better suited. Those are called adaptations. One misconception is that adaptations are always good. They're not necessarily always good, but if they're a beneficial adaptation, then they'll help that organism survive in that particular environment. And when it comes to evolution, that's the process of change that life has changed as time has gone on. Sort of the culmination of multiple adaptations with time. Now, biology as a broad study, it is the scientific field of life in general. And essentially any questions that could be asked about what life is, how life occurs, or what occurs in living organisms, down from cellular all the way up to multicellular organisms as large as whales. That is what biology is meant to cover. And so life in general, it can range from a broad scope from flowers, rabbits, seahorses, giraffes, butterflies, insects, mammals, and plants and cells. Essentially what we were just covering is when defining life, what are some basic characteristics that are used to identify something as living? Is there some order or structure to how it is built or consisted? In the case of multicellular organisms, how do their cells come together to form higher structures? Evolutionary adaptations, essentially, do these organisms change with time to best fit their environments? Is there some sort of regulation with how materials are used to allow that organism to continue living? Do they reproduce? Whether asexually or sexually, does that current organism attempt to make more of itself further with time? And is there a certain response to the environment? Certain stimuli, whether food in the area or certain dangers, does that organism change in some way, shape, or form? Whether as simple as does the day become night, or is there just some change in the environment that results in an organism responding to it? And then growth and development is necessary in life for it to change, to develop with time. Now, naturally, if you take in nutrients, resources, you need to be able to process. Life should process energy in order to do those characteristics. Now, study of life, it involves plenty of themes. Now, biology, ranging from the microscopic to the macroscopic, it covers a wide variety of life or factors that affect life. But there are 5 unifying themes, which include how life is organized, what information is present, the energy and matter that's used for life, the interactions between different organisms or their cells and their environment, and how they change or how they evolve with time. Now, taking all these into effect, we can order life into various levels, ranging from the molecular all the way to an entire living being, or all the way up to planetary. This enormous range is broken up into various biological levels. Going from large to small, we have the biosphere, which could be the sum total of life on a particular planet, which so far we just look at Earth. Ecosystems, the different types of environments that could be seen, or communities of organisms in their environment. The different individual populations, and then to a particular part of that population, an organism. Then we can look at if there are organs present and how those organs are made from tissues and how each individual tissue is made from cells. Cells in multicellular organisms, or I should say eukaryotic cells, they have organelles, which are like organs for cells. And then going further, organelles are inevitably made from molecules, which come from atoms. This is just different examples of those different ways of ordering life, from giant groups of life all the way to particular individuals and what constructs those individuals. Emergent properties, those are what result from the arrangement and interaction of parts within a system. Emergent properties characterize non-biological entities as well, not just living. For example, a functioning bicycle emerges only when all the necessary parts connect in the correct way. Sort of like how when a certain protein is being made, it's made from different amino acid components, and then once they all come together, then there's a protein that provides a particular function, whether building certain tissues in our bodies or allowing for certain mobility from cellular to macroscopic. Now, the reductionist approach, that studies the isolated components in a living system. To explore emergent properties, biologists complement reductionism with systems biology, which is the analysis of interactions among the different unique parts of a biological system. For systems biology, systems biologists can study life at all levels. When it comes to structure and function, at each level of a biological hierarchy, there's a correlation between how it's structured and what functions occur from those structures. Analyzing a biological structure gives clues about what it does and how it works. Conversely, knowing the function of something provides insight into its structure and organization and might help understand what a similar structure in a different organism does if that organism is not as well studied. Here we have hummingbirds. They have the ability to suck nectar from flowers, having wings as well that help to float around. Now, going all the way down to a cell, an organism's most basic unit of life that both has the properties and ability and all different parameters needed for life. The cell being the smallest organization that can perform all the activities of life, every cell is enclosed by a membrane, a phospholipid membrane that regulates the passage of materials into and out of the cell, between it and its environment. The cells of bacteria and archaea are prokaryotic, while all other forms of life are composed of eukaryotic cells. A eukaryotic cell, it simply has a nucleus organelle where its genetic material is stored, whereas for a prokaryotic cell, its genetic material is not stored in a nucleus, but is floating around in its cytoplasm, which makes it more susceptible to changes from its environment, such as UV radiation or what have you. Looking at a typical eukaryotic cell, there are multiple organelles. Some particular parts to notice is we have our membrane, our membrane, which surrounds our cytoplasm, the liquid inside a cell. Multiple organelles that have various different functions, and we'll get into those. Prokaryotic cells, the main difference being its genetic material is not stored inside an organelle, but it still has a membrane. These are typically much smaller, on a much smaller scale than eukaryotic cells. Like processes involve the expression and transmission of genetic material. Within cells, structures called chromosomes contain the genetic material in the form of deoxyribonucleic acid, DNA. And this is just an example of a cell that has multiplied its DNA to 2 times the copies. And then those 2 cells start dividing to form, or that one cell starts dividing into 2 new cells in order to asexually reproduce. Now, the genetic material, DNA, each chromosome contains one long DNA molecule with hundreds of thousands of genes that encode different functions and traits for an organism to be able to do. Genes are the units of inheritance. They encode information for building the molecules that our cells produce. The genetic information encoded by DNA directs the development of an organism to the point where it starts exhibiting different traits or produces organs that have particular functions. In sexual reproduction, that is when genetic material from more than one source comes together. Looking in the case of animals, a sperm cell with half the genetic material used to produce an organism of that species meets an egg cell that also contains half the genetic material needed to make a whole organism. They come together in a process called fertilization, and then that fertilized cell reproduces into multiple cells that eventually form that organism as a whole. And to get further into it, while the sperm and egg cell have half the genetic material to make an organism, the fertilized egg has an exact copy to make a whole organism. Therefore, each cell afterwards will have all the same genetic material as every other cell in that specific organism. The molecular structure of DNA accounts for its ability to store information. Each DNA molecule consists of 2 very long strands that are wrapped around each other in a repeating double helix, essentially taking 2 wires and twisting them continuously over each other. Now, each individual chain is made up of a combination of 4 kinds of building blocks called nucleotides. The 4 types are adenine, guanine, cytosine, and thymine, AGTCT. Now, when you're making one strand of DNA, the order of those nucleosides are what help determine different genes further down the line. Now, while a single strand contains nucleotides, DNA consists of 2 of those strands that are wrapped together. And what we're seeing is that certain nucleotides on one strand like to pair with certain nucleotides on the second strand. For example, anytime you see a cytosine on one strand, it will like to join up with a guanine on the second strand, or the guanine on the first strand likes to pair up with cytosine on the second strand of DNA. And likewise, thymine and adenosine like to base pair as well. For many genes, the sequences provides the blueprint for making a protein eventually, and therefore protein encoding genes control which proteins are made, but they do so indirectly. What first happens is that DNA, deoxyribonucleic acid, is first transcribed into ribonucleic acid, RNA. Now, this RNA is then translated to make proteins. This is beneficial in eukaryotic cells in that since our DNA is stored in a nucleus, that important genetic material is protected, whereas the RNA can be made from DNA, leave the nucleus, and then be exposed to the dangers that could have changed DNA. But RNA can then be used to make protein. And this is involved in the process of gene expression, which is converting genetic information from a gene into a cellular product like proteins. Now, the whole process is called central dogma, which is starting from transcription, making messenger ribonucleic acid from DNA, and then through a combination of messenger RNA, transfer RNA, and ribosomal RNA, translation occurs where a series of amino acids are combined together to eventually form a whole protein. And then once that protein folds into a unique three-dimensional structure, it then has the function that that protein is needed to do. Genomics on a large-scale analysis of DNA sequences. An organism's particular genome is its entire library of genetic information. Genomics is the study of sets of genes in one or more species and what those genes particularly do. Proteomics is the study of the whole set of proteins that an organism or species will make and what properties those proteins have or perform. The entire set of proteins expressed by a given cell tissue or organ is called the proteome, such as the entire genetic material is called the genome. The genomics approach depends on high throughput technology, in other words, intensive measures that yield enormous amounts of information. Bioinformatics is the use of computer technology and biological information that combine to help look through large volumes of data to glean out repeating patterns or repeating functions in genes or proteins. Now, the theme that life requires the transfer and transformation of energy and matter, the input of energy from the sun and the transformation of energy from one form to another makes life possible. Life, or I should say energy in particular, a law from thermodynamics and physics and chemistry is that energy cannot be created nor destroyed. Therefore, energy must transfer from one source to another. And on life on Earth, all of our energy starts from the sun. The chemical energy generated by plants and our photosynthetic organisms, are called producers, is first formed by taking energy from sunlight, and this is passed to consumers, which are organisms that feed on other organisms to get that sunlight energy. When organisms use energy to perform work, some energy is lost to the surroundings as heat, which is simply wasted energy that could not be used by that organism for something beneficial. As a result, energy flows through an ecosystem, usually entering as light and exiting as heat, and slowly down the cycle from producers to consumers. More energy is lost than was going from one level to next than was originally obtained. Chemical cycles within an ecosystem, those are where energy is used and then recycled in some way. Now, from molecules to ecosystems, interactions are important to biological systems. Interactions between the components of the system ensure smooth integration of all the parts. This holds true equally well for components of an ecosystem and the molecules in a cell. Now, interactions between organisms, between the components of an organism include the interactions between organs, tissues, cells, and molecules that make up living organisms. to have a smooth functioning life form, many biological processes can self-regulate through a mechanism called feedback. In feedback regulation, the output or the produced product of a process regulates the very process. For example, if a lot of a certain protein is made, then the organism might then stop producing more of that protein until there's not as much left. The common form of regulation in living organisms is negative feedback, which is the response reduces the initial stimulus. So a less common form of regulation is positive feedback, in which an end product speeds up its own production. An example of negative feedback is insulin producing cell in a pancreas in response to a large amount of glucose in the blood. High blood glucose stimulates the pancreas to release insulin. Insulin then circulates throughout the body and binds to body cells, causing them to absorb glucose, and liver cells to store more glucose than they were. This results in lowered blood glucose, and the lowered blood glucose no longer stimulates insulin secretion. An ecosystems and organisms' interactions with other organisms and the physical environment. At the ecosystem level, each organism interacts with other organisms. These interactions may be beneficial to harmful to one or more organisms. Organisms also interact continuously with the physical factors in the environment, and environment is affected by the organism living there. In this case, each organism interacts continuously with physical factors in its environment. Humans interact with our environment sometimes with dire consequences. Over the past 150 years, there's been a sharp increase in the presence of carbon dioxide in the atmosphere, which, like many gases, has the insulating effect of retaining more heat that is received from the sun than what is released. Wind and precipitation patterns are also changing due to this changing capture of heat. Extreme weather events such as storms and droughts are seen more often than in past recorded history. As habitat deteriorate, plants and animal species shift their ranges to more suitable locations. Therefore, populations of many species are shrinking in size or even disappearing. Now, evolution accounts for the unity and diversity of life. Evolution is the one idea that makes logical sense of all different factors we know about living organisms. Scientific explanation for both the unity and diversity of organisms is evolution. The concept that living organisms are modified descendants of common ancestors. An abundance of evidence supports the occurrence of evolution, i.e., are you somewhat different from your ancestors? Nothing in biology makes sense except in the light of evolution. A quote from Theodosius Dostoevsky, a biologist. Now, classifying the diversity of life, approximately 1.8 million species have been identified and named to date. Each species is given a two-part name to help identify it from other species. There is the genus, to which the species as a whole belongs, and then a species name unique to that specific species. Multiple species can exist in a certain genus. For example, we have Homo sapiens, the name of our particular species of human. while in the past there were other types of humans like Homo erectus or Homo neanderthals. Estimates of the total number of species that actually exist range from 10 million to 100 million, though likely there are countless that we simply will never be able to either observe or factor in from the sharp range of microscopic to macroscopic life. Life is currently broken into 3 domains. Those 3, how all life on Earth is broken up into, starts out as bacteria, archaea, or eukarya. Prokaryotic life forms are in bacteria and archaea specifically, whereas multicellular organisms, or I should say eukaryotic organisms, are eukarya, so anything that has a nucleus. Domain eukarya includes the protists and the following 3 kingdoms: plants, which produce their own food via photosynthesis, fungi, which absorb nutrients from decomposing matter or other organisms, and animals, which ingest their food through consumption. Protists are more closer to single cellular, but eukaryotic organisms nonetheless. The most numerous and diverse eukaryotes are the protists. They are mostly single-celled. They are classified into several groups. Some protists are less closely related to other protists than they are to plants, animals, and fungi. So breaking everything down, we have bacteria and archaea. Archaea are typically thought of as extremophiles. You'll see those, they can be observed in high conditions that usually other life cannot live in, either high salt content, high heat, high methane gas from oxygen or other typical atmosphere. And then in the domain of eukarya, there's the kingdoms of plants, fungi, protists, and animals. The 2 unique names being plantae and animalia. Unity in the diversity of life. A striking unity underlies the diversity of life. For example, DNA is the universal genetic language common to all living organisms, and unity is evident in many features of cell structure. The history of life as documented by fossils and our evidence is the saga of a changing Earth with an expansive amount of time. Some common structures range from cilia, which can help unicellular organisms move around or microscopic organisms move around, or as present in the breathing pipes of animals, they are used to help move objects that get into the windpipe out of the windpipe to either be thrown up or to go down the esophagus into the stomach. So using similar structures, but in different ways. This would be an example of unearthed fossils. Charles Darwin and the theory of natural selection. This was proposed in his original book, The Origin of Species by Means of Natural Selection, in 1859. Darwin made 2 main points from his observations. Species showed evidence of descent with modification from common ancestors, as generations change, so too does that population of an organism from its original ancestors. Natural selection being the second point, which is the mechanism behind descent with modification. Essentially, if through mutation or inheritance, an organism has a trait that makes it more likely to survive in its environment, then by probability, that trait will have a higher likelihood of being transferred to the next generation, because that population will be more likely to reproduce later on. Darwin's theory explained the duality of unity and diversity. Thing to note, The Origin of Species is a book from quite a long time ago. Its value is that it proposed these new ways of thinking, though there has been much advancement from the original ideas proposed, as our understanding of genetic material down to DNA structure has occurred. One example being, if birds came from some common ancestor, with time, different mutations or genetic traits have been inherited, such that we have pink and blue flamingos, to different types of robins, to flightless birds like penguins that are more adept at swimming. And a personal side, the change from flamingos with bendy legs to penguins with not so much bendy legs. Now, Darwin observed that individuals in a population will vary in their traits. Many of those traits seem to be heritable. More offspring are produced, then survive. Therefore, there is competition between the resources available in that environment. Species generally suit their environments. Darwin reasoned that individuals that are best suited to their environment are more likely to survive long enough to reproduce. Over time, as these factors compound over and over again, more individuals in a given population will have that advantageous trait. Evolution occurs as the unequal reproductive success of individuals. The natural environment selects for the propagation of those traits, which it should be mentioned that evolution does not always result in beneficial traits, but the ones that are beneficial are what help an organism survive. Now, Darwin called this particular process of beneficial traits having a better likelihood of being passed on as natural selection. For example, a population such as beetles with different outside colors, perhaps some colors seem more delicious to a predator, therefore that would be a disadvantageous trait, and the remaining individuals without that trait or a different trait are more likely to survive and reproduce, resulting in a higher frequency of the advantageous version of those traits. Natural selection results in the adaptation of organisms to the circumstances of their way of life and their environment. For example, bat wings are an example of adaptation, aiding those mammals to actually fly. The shared anatomy of mammalian limbs between different types of mammals reflects the inheritance of the limb structure from a common ancestor. Fossils provide evidence of anatomical unity from descent with modification. For example, as we look at bat wings, what it looks like is similar to many mammals that have hands and fingers. It appears that those wings are really just very, very long fingers with webs in between. Darwin proposed that natural selection could cause an ancestral species to give rise to a different species from the original, or sometimes more than one descent species. For example, the finch in the Galapagos Islands are descended from some common ancestor, but some finches have hard beaks that are strong enough to crack nuts, and some have longer, slender beaks that are easier at reaching bugs that are further within a tree. Evolutionary relationships are often illustrated with tree-like diagrams that show ancestors and their descendants. For example, some common finch that over time broke up into different species with time, and those different species have different adaptations or collections of compounded traits that allow them to obtain different resources through various success from their environment, such as different finches that either eat insects or fruits, or those who actually prefer seeds, or those who are able to get into cacti. In studying nature, scientists make observations and form and test hypotheses. The word science is derived from Latin, which means to know, and inquiry is the search of information and explanations of natural phenomena. Biologists use a process of inquiry that includes making observations, forming logical hypotheses, and testing them. Explanation and observation. Biologists describe natural structures and processes. This approach is based on observation and analysis of data. Recorded observations are called data. Qualitative data often takes the form of recorded descriptions. Quantitative data are expressed as numerical measurements, organized into tables and graphs that can be gleaned. I believe this is Jane Goodall, famous biologist who studied many chimps and monkeys. This is an example of her notebook gleaming information. Inductive reasoning is how we draw conclusions through the logical process of induction. Repeating specific observations can lead to important generalizations. For example, the sun always rises in the east, or all organisms are made of cells. In science, a hypothesis is an initial explanation based off of initial observations and some assumptions that leads to a testable prediction. It must lead to predictions that can be tested by making additional observations or performing experiments. Now, an experiment is a scientific test carried out under controlled conditions. For example, an initial observation says or shows your desk lamp doesn't work. A question that arises might be, why does the lamp not work? And two potential hypotheses that, as a hypothesis, are testable. The bulb could possibly not be screwed in properly, or the bulb is burnt out. And both of these hypotheses are testable. For example, reinstalling the bulb could potentially fix it, and if that does fix the issue, well then that's likely what caused it. If the lamp does not turn on after being screwed in, or screwed into another outlet, then likely the lamp is burnt out instead. Deductive reasoning uses general premises to make specific predictions. Initial observations may give rise to multiple testable hypotheses. We can never prove that a hypothesis is completely true, but testing it in many ways with different sorts of data can essentially increase our confidence that that hypothesis is likely what is happening. Something that should be noted, hypotheses are specifically to test what is happening in our physical world. Supernatural and religious explanations are not what science is meant to prove or disprove. Science is only meant to be confined to the natural world and explain with the current available resources what might be happening, or what could be an explanation that focuses just on the physical world. The scientific method is an idealized process of inquiry. However, very few scientific inquiries adhere rigidly to this approach. Backtracking or rethinking may be necessary partway through the process. For example, when you're forming and testing a hypothesis, you may end up finding different outcomes, or you may need to which may lead to different feedback. And over time, your initial hypothesis might need to be changed as you're doing different tests, and eventually you form one that seems to stand the test of time. Initially in the scientific process, there's forming and testing a hypothesis based off of initial observations, and will require performing tests that involve interpreting data later, that either support or contradict the hypothesis, and which may inspire a revised or new hypothesis. Exploring and discovery is essentially observing further information in nature, asking questions with what's being observed, and or reading the scientific literature available. Now, what can happen is that societal benefits and outcomes occur, such as developing technology, informing policy, governmental, health, care, insurance, and beyond, solving current problems and issues, or past problems, and building knowledge. Community analysis and feedback essentially is called the peer review process, where those knowledgeable in a field look through current research to determine if it was produced to high standards worthy of trust, or if those findings can be reproduced, which further supports that the claims made can be held in confidence, eventually leading to publication and a consensus building. A case study in scientific inquiry investigating coloration in mouse populations. Color patterns of animals vary widely in nature, sometimes even between members of the same species, such as with dogs and cats, or even different types of Pomeranians, going from gold to brown to black to white. Two populations of mice belonging to the same species, Paramystis polionotus, but with different color patterns, are found in different environments. The beach mouse lives on white sand dunes with sparse vegetation, whereas the inland mouse lives on darker soil. Therefore, the beach mouse is better able to hide from its predators due to being able to blend in with the surrounding, whereas the inland mouse is better able to hide from its predators by not standing out. Therefore, likely, if both populations occurred in the same area, one population would be less likely to survive and reproduce in its current environment than the other. The two types of mice match their coloration to their habitats. Natural predators of these mice are all visual hunters. Therefore, it was hypothesized that the color patterns had evolved as a culmination of adaptations to protect the mice from predators. Not that these adaptations were done with thought, but whether mutations along the line resulted in this color to form. Those with that mutation were better likely to pass along that trait to the next generation. Hopska, and a group of students tested this hypothesis. And the researchers predicted that mice that did not match their habitat would be preyed on more heavily than mice that did. And they built models of mice, painted them to match one of the surroundings, and placed equal numbers of each type of model, each habitat, and then recorded the signs of predation. Now, in the beach habitat, where it's lighter colorations, the percentage of attacked models increased with those that did not fit the lighter background, whereas those inland, the lighter coloration, were less camouflaged and more likely and more found to be predated. Here what we have different populations. There will always be in a particular experiment a control population and an experimental population. The control being one that no changes have been made in the typical location, whereas the experimental is where a trait was changed to test if that changed trait leads to a observable change in the environment. For example, how often mice were attacked. In a controlled experiment, experimental group is compared with a control group. Experimental variables are features or quantities that vary in an experiment from the control. The independent variables is the one that is manipulated by the researchers, while the dependent variable is the one predicted to be affected in response. Independent variable is what's changed, whereas the dependent variable changes based off of the independent variable. In the context of science, a theory is a broader scope than what a hypothesis covers. They are general and can lead to new testable hypotheses and are supported by a large body of evidence in comparison to a single hypothesis. Most scientists work in teams, which often include graduate and undergraduate students, those going for their master's and doctorate degrees, alongside those going for their associate's and bachelor's degrees or certificates at any level. Good communication is important in order to share the results through seminars and publications and websites. Scientists check each other's claims by performing similar experiments. If experimental results are not repeatable, the original claim will need to be revised due to a lack in confidence of it. It is not unusual for different scientists to work on the same research question. Scientists cooperate by sharing data about model organisms, for example, the fruit fly Drosophila melanogaster. The goal of science is to understand natural phenomena. The goal of technology is to apply scientific knowledge for some specific purpose, whether to make a product more easily to sell or to make a healthcare technology that can better heal or treat wounds without leaving permanent marks. Biology is marked by discoveries, while technology is marked by inventions. The combination of science and technology has dramatic effects on society. For example, the discovery of DNA by James Watson and Francis Crick allowed for the advances of DNA technology, such as testing for hereditary diseases. Now, just to go further, someone of note of importance in the discovery of DNA who is often overlooked, is Rosalind Franklin, who essentially made many notes on genetic studies that James Watson and Francis Crick then used in part of their overall consensus discovery. James Watson and Francis Crick built up a body of information, but a good portion of that was based off of Rosalind Franklin's initial findings. Now, there are debates on technology that center more on should we do it than can we do it. This involves ethics. The combination of science and technology has dramatic effects on society. For example, the discovery of DNA. Let's see. Ethical issues can arise from new technology, but have as much to do with politics, economics, and cultural values as with science and technology. So there are numerous factors that go into how life progresses on. The value of diverse viewpoints in science. Many important inventions have occurred where different cultures and ideas mix. For example, the printing press relied on innovations from China, such as paper and ink, and Europe, such as the mass production in mills. Science benefits from diverse viewpoints, or I should say from diverse views from different racial and ethnic groups, from both men and women. The reason being, depending on the valuable factors that go into building who you are as a person, may incline you to think different ways. Certain communities might have an easier time thinking of different solutions, whereas certain communities might have a better time thinking of other solutions, or may have a better familiarity with thinking certain ways. Therefore, the culmination of these diverse views helps produce better and better results. The more voices heard, the more robust, valuable, and productive the scientific interchange is, and therefore we're able to get a wider range of ideas, some of which will need to be removed with time versus some that can then be brought into a greater consensus. These are some of the figures. And that concludes Chapter 1 of General Biology. Glad to have you all.